So then, we start with part 2, our epigenetic toolbox, the introduction into epigenomic data analysis. Actually, we will not talk so much about the data analysis part because I think this is a bit beyond the scope of this webinar. But I will rather focus on the methods that we perform in the lab for the genome-wide analysis of the epigenetic modifications, but I will also show what we can expect as output. And in the end, I will also do a short highlight of some data analysis tools and strategies. Okay, so we've already seen that there are really a lot of different epigenetic modifications. And of course, there are also a lot of different methods. Today, I just want to show several, I would say, the most important or the most favorite methods which are widely adopted in the community. And of course, for each of these methods, there are a lot of different protocols out there with a lot of different versions. But I will just focus on the principles of the methods. So first of all, I would like to talk about ATAC-seq for the analysis of chromatin accessibility. Then about ChIP-seq, this is a method where we can profile histone modifications, but also transcription factors which are bound to the chromatin. For DNA methylation analysis, I want to show two different experiments, whole genome Bisulfite sequencing and reduced representation Bisulfite sequencing. And finally, as the transcriptome can be seen as the functional readout of the epigenome. And we frequently also study this in epigenetic studies. I will also say a few words about RNA sequencing. Okay, but let's start with chromatin accessibility analysis. So ATAC-seq. ATAC is short for the assay for transposase accessible chromatin. And as the name already implies, it relies on a hyperactive transposase, the TN5 transposase. And the beauty of this approach is that this transposase is already preloaded with sequencing adapters. So the adapters that are later on needed for binding to the sequence's flow cell that will allow the sequencing process. So now at the beginning of the protocol, you need to extract intact nuclei. This is actually the major challenge of this protocol, depending on tissue or the samples that you want to use. And then you simply incubate your nuclei with this transposase. And now the transposase can only access regions where the chromatin is or the DNA is accessible. So this means it will only go into regions where the nucleosomes are shifted apart from each other. And these are the accessible regions, the euchromatic regions. It cannot bind directly to the nucleosomes and also not to regions where the nucleosomes are very closely together. So the transposase will then bind and it will simultaneously fragment and tag the DNA, which is accessible. This process is then called tagmentation. And we end up with DNA fragments, which already have the sequencing adapters attached to each side. So now we can then purify these DNA fragments and enrich them by PCR, and then simply put them on the sequencer. As readout, we will then get reads which are enriched in the accessible regions. So in a genome browser, this looks like this. Now this is a very large region, 2000 KB, but we can already see that there are regions where we have peaks. So these are open regions or accessible regions. And we also have regions where we do not have peaks. So these are closed regions. And if we look closely, for example, here, you can already see that there are also differences between the samples. So for example, in this sample, this region is open. And in this sample, there's hardly any signal. So it is more closed. And if we now zoom into one of these peaks, then we can see, for example, this is an actively transcribed gene. And here at the promoter region, we have a strong ATAC signal. So this is a accessible and probably active promoter. So then we come to ChIP-seq, the method to profile histone modifications and also for transcription factor binding analysis. ChIP-seq is short for chromatin immunoprecipitation. And here the principle is that we prepare our chromatin, so we isolate it from the cells. Then in some way we have to fragment the chromatin. For this we can use sonication to shear the chromatin into fragments, or we can use enzymes or also transposase. And the key to this method is that then we use a specific antibody which will bind to our modification of choice. So for example, again, my favorite histone modification, H3K4 trimethylation, we now have an antibody which specifically will only bind to nucleosomes which have this modification. And then in the next step, we can use the antibody, which is bound, for example, to a protein G bead, to extract only these nucleosomes which have our modification. And then we extract the DNA and prepare a sequencing library and put it on a sequencer. So if we do this now for different histone modifications, we will get different genome-wide histone modification profiles. And now this is an example how the histone modification landscape looks at a typical active promoter. Here on top, we have again the ATAC-seq that we already saw before. And what's quite interesting is that here, directly at the ATAC-seq, there were no histone modification signals. And this is because here, this ATAC-seq is actually a nucleosome depleted region. This means that it's so accessible that the nucleosomes are shifted to the sides of this region. And if there are no nucleosomes, we can, of course, also not have histone modifications. But directly neighboring this ATAC-seq, we have a strong enrichment of the active histone modifications H3K4 trimethylation and H3K27 acetylation. So these are very prominent promoter marks. What we also see is H3K36 trimethylation, which is enriched along the gene body of the transcribed gene. So this is a good indication together that this gene is really transcribed. And below here, the repressive marks, they are not enriched. So, but to get a picture like that, we have to run an experiment for each of these histone modifications. Okay, so both ATAC-seq and ChIP-seq were enrichment-based methods. So where we only sequence the reads, which are active, which are accessible, or which have the specific histone modification. And for DNA methylation, we use a very different approach. The gold standard and also the essential part of, I would say, all of the modern DNA methylation analysis for genome-wide profiling of DNA methylation is Bisulfite conversion. So to understand what that is, we need to have a look at some chemical structures. So if we have an unmethylated cytosine and we treat it with sodium bisulfite, then we have a sulfonation reaction. This means we get here the sulfate group and we end up with cytosine sulfonate. In the next step, because this is quite unstable, we have a spontaneous deamination and we end up with a uracil sulfonate. So we do not have cytosine anymore, but uracil. And if we then add NaOH, so we increase the pH, then the sulfate group is lost again, and we end up with uracil. So this bisulfite conversion means that we convert unmethylated cytosine to uracil. And the important part is now, if we have 5-methylcytosine, so methylated DNA, then this first step of the bisulfite conversion cannot happen. And this means after bisulfite conversion, methylated cytosine is still cytosine. Now we can make use of that for our analysis. We already know that cytosine pairs with guanine, but uracil, which is the RNA equivalent to thymine, pairs with adenine. So if we use our DNA in a bisulfite conversion and then perform a PCR on that, we will in the end get, so uracil will then again be a thymine because after PCR we have double-stranded DNA. And we can make use of that because in the unmethylated DNA sequence, the cytosines are converted to thymines, while in the methylated DNA sequence, we have no conversion. So let's just look for illustration at this example sequence. So here we have a CG, then here we also have a C outside of a CG context. So this is definitely unmethylated. And here again a C and also a CG. If the sequence now is unmethylated, then all of these Cs will be converted to Ts after the PCR. So we have a completely altered DNA sequence of the bisulfite DNA. But if these CGs are methylated, only in the CG context, they will stay CGs. But the Cs which are not in a CG context, they are unmethylated, so they will be converted. And now if we just take these DNA sequences after bisulfite conversion and sequence them, we can compare them with the genomic sequence and then see, okay, there was a conversion. So this has to be an unmethylated cytosine. Or we see here, oh, there's no conversion. So this was a methylated cytosine. And there are several methods which make use of this. The gold standard for DNA methylation analysis is whole genome bisulfite sequencing. Again, there are different protocols, so this is just one that we use frequently in the lab. You take your genomic DNA, you incubate it with the same transposase, which I already described for ATAC-seq. It inserts the sequencing adapters. You have to do a gap repair to get covalently bound adapters on both strands. And then the important step, you introduce the bisulfite treatment. So you will have the conversion of unmethylated cytosines. Then you enrich it by PCR and you can sequence it. As readout, you get something like this. You will have reads on across almost the whole genome. And now you can go to the CpG positions and you can read out what is a T and what is a C, so what is methylated and what is unmethylated, and then calculate the percentage of methylation on each CpG for the cells that you analyzed. So with that, you can cover almost all of the CpGs in the genome. So I think there are 28 million CpGs in the human genome. But the problem is for that, you have to sequence the whole genome with a high coverage. And this is expensive. And on top of that, you will also get a lot of uninformative reads, because I already explained in part 1 that CpGs are not randomly distributed across the genome, but they are enriched in certain regulatory regions. So we will sequence a lot of the genome, which doesn't give us any information. So a method which tries to reduce the costs is reduced representation bisulfite sequencing. Here we make use of the fact that CpGs are often enriched or mostly enriched in CG-rich regions. So we can use restriction enzymes, which preferentially cut or specifically cut in CG-rich regions, and then extract just these parts of the genome. So for example, here we have HAY3. This is an enzyme which cuts between the sequence GHGA. Here it is, it will cut independently if these Cs are methylated or not. This is of course very important for this approach, so that you do not get a methylation bias by this choice of enzyme. So we incubate our DNA with H3. It will cut in these CG-rich regions, and then we extract only the small fragments and prepare a sequencing library from that. By that, we will get the following readout. We will not get a coverage of the whole genome, but we only will get coverage between the cutting sites. Using H3, we can cover about 5 million CpGs in a human genome. So, of course, by this method, we lose a lot of information, but also the sequencing costs are significantly lower. So depending on your experimental design, it often makes sense to also use RRBS as an alternative for whole genome bisulfite sequencing, especially because the CpGs which are enriched are often in very interesting regions in the CpG ions, promoters, or enhancers. Okay, so if we now did the sequencing, this is now an example of the readout that we get. This is now from whole genome bisulfite sequencing. It looks similar for reduced representation bisulfite sequencing, just with lower coverage across the genome. And so the color code here means blue is unmethylated and red are methylated CpGs. And if we focus here on this region, then we can see in the upper sample, this is mostly unmethylated. So these are many CpGs next to each other. So this is an CpG island. And in the second sample, you can see that this is partially methylated. So this will most probably have an effect on the expression of this gene. In this upper sample, this gene is probably transcribed, and in the lower sample, probably silenced. Okay, so then finally, a few words on RNA sequencing. Usually when we talk about RNA sequencing, we are mostly interested in the messenger RNA, the mRNA, because we want to study which genes are expressed as proteins. And again, a lot of different approaches, but what they mostly have in common is that they make use of the poly-A tail of the ripe mRNAs. Because of this poly-A tail, you can use primers which have an oligo-DT stretch, and this can then bind to the poly-A tail of the mRNA, and by using beads or PCR or whatever, you can then pull out or enrich specifically the mRNA, and then prepare cDNA from this. And based on the cDNA, you can again prepare a sequencing library and go for sequencing. As we pull out the mRNA based on the poly-A stretch, we mostly analyze ripe mRNA. So this is already spliced. And this means that most of the reads will map to exons, and we will get little intronic reads. Okay, so this is all I wanted to say about the methods. And now just a few remarks on epigenetic data processing. So when I was preparing for this webinar, I was wondering what to show and what is really important to have in our epigenetic toolbox. The problem is, we already saw that there are a lot of methods, but there are, of course, far more methods for data analysis. So on this website above here, you can see this is just a collection of some analysis tools which are widely used in the community. And of course, we cannot go through all of them. So if you want to do some further reading, have a look on this website. So what I wanted to say instead is that if you want to do epigenetic data processing, a very good start are the NF-core pipelines. So NF-core pipelines, you might already be familiar with that. They rely on a community effort to generate next flow pipelines, which come as Docker or Singularity containers, so they are very easily to install. And the very big advantage is that they are highly reproducible and standardized, And they also have a great documentation. So it's really easy to, to start working with that. And during the last month or last year, our lab also switched completely to using these pipelines for data processing. And they are already available for almost all types of epigenetic data that we can imagine. So as an example, and I will not go into details, here for ATAC-seq data analysis, you can use the NF-core pipeline for data processing, but also for several downstream analysis. You can really start with your raw reads directly with the FASTQ files. You will get a QC, you can trim the adapters, then you can choose one of your favorite aligners to align the reads to the genome, mark duplicates, and you can do all kinds of different things to do exploratory analysis. For example, you can call peaks, so regions which have accessible chromatin. You can annotate these peaks to genomic features. So for example, you can check how many of these open regions are enhancers. And you can also generate the tracks that I visualized before in the genome browser. So, and you can even do differential analysis between different samples. So this is a really handy pipeline for that. A similar pipeline is also available for ChIP-seq. So the tools are very, almost the same. So I do not show them here. You can also, use this pipeline for ChIP-seq peak calling. So, defining the regions where your favorite histone modification is enriched. Do a very extensive QC and also do differential analysis between different sample groups. Of course, there's also a pipeline for RNA sequencing. Again, you start with your FASTQ files. You can do several steps of pre-processing of the data. Align again with one of your favorite aligners, the reads to the genome or to the transcriptome, and then prepare also files for further downstream analysis and go for differential analysis. So for DNA methylation, there I think data analysis is a bit more complex than for the other data types. Here we usually use the method-seq pipeline now for data processing. There are two different workflows, but just let's stay with the Bismarck workflow. So we start with the FASTQ files, we do QC, we trim the adapters, we align the reads, and now importantly, we in the same step can also extract the methylation codes. And these methylation codes are then usually used for extensive further downstream analysis with other tools. So in the interest of time, I will not show many more other tools for downstream analysis. I just want to advertise RNA-seq because this was co-developed by our lab. So if you're interested in going into DNA methylation analysis, you might check it out. The principle is that we can start with the methylation calls from the NF-core method-seq pipeline to put it into RNA-seq. Then we can get much more extensive quality control measures. We can pre-process the data further. We can infer covariates. We can also do exploratory analysis, which are always very helpful. And we can also do differential methylation analysis. And we can generate more tracks and tables for further data handling or for visualization also in a genome browser. Okay, so with that, I hope that you got an impression of of the different methods that we need to come to such a picture. So by the way, this was this picture comes from the cell line HepG2 and was here all the different assays were run on the same sample, and this was done during the German Epigenome program in our lab several years ago. And I really like this figure because it sums up things very nicely. So for example, here you can see very tiny. So in red here, this is methylated DNA. And in green, this is unmethylated DNA. And here between the methylated parts, there's a very small unmethylated promoter region. And exactly fitting to this, there's also enrichment of the active histone modifications, H3K4 trimethylation and H3K27 acetylation. The gene body of this gene shows also H3K36 trimethylation, which is found in transcribed gene bodies. And if we look here at bulk or single cell mRNA expression, we also see that this gene is transcribed. So it sums up things very nicely. An active gene shows this epigenetic landscape, and this goes together with transcription. Or if we look here, then we have repressive marks, and we do not have gene expression of these genes. So this is a silenced region. Okay, so this was already the summary. Here are again some of the resources that I showed already during part 2. And now I'm happy to take some more questions.